Good morning all. I am Srishna K, first MSc Biology student, St. Mary's College, Thrissur. So today we are going to deal with the topic DNA damage. DNA damage is any kind of alterations in chemical or physical structure of DNA molecule such as base missing, strand breaking, etc. DNA damage get by many ways such as ionizing radiation, chemical exposure, UV exposure, replication errors, etc. DNA damage induces abnormalities in DNA molecule and it will not be able to work well. DNA damaging agents. It include biological, physical as well as chemical agents. Mainly they are UV radiations, ionizing radiations, oxygen radicals, replication errors, reactive oxygen species, chemotherapy, chemical exposure, etc. Difference between DNA damage and mutation. DNA damage is physical or chemical abnormalities in DNA, whereas mutation is changes in base sequence of DNA. DNA become non-viable if a DNA got damaged. In case of mutation, DNA is viable. No flow of information in DNA damage, but in case of mutation, flow of information occurs. In most cases, DNA damage can be repaired. Mutation cannot be repaired. Types of DNA damage. Single base alterations. They are depurination, deamination, alkylation, base analog incorporation, mismatch bases. Double base alterations. Purine dimer and pyrimidine dimer. Chain breakings. Single-stranded break and double-stranded break. Cross-linking between DNA and DNA, between DNA and protein. Single base alterations. Depurination. It involves the loss of purine bases adenine and guanine from DNA. The major causes are UV radiation, ionizing radiation, free radicals, chemical induced like estrogen, monofunctional alkylating agents. It was by the spontaneous hydrolysis of N-glycosyl linkage and it produces an abasic site in DNA. All the hydrolytic reaction result in alteration to DNA that are unnatural. Example, depurination of guanine by hydrolysis create apurinic deoxyribose. Deamination, single base alteration, hydrolytic removal of an amino group from DNA. Major causes are spontaneous and chemically induced. Example, deamination of cytosine result in formation of uracil. Deamination of adenine result in formation of hypoxanthine. Deamination of guanine result in formation of xanthine. Deamination of 5-methyl cytosine result in the formation of thymine. The structural representation of spontaneous deamination is shown here. Alkylation, single base alteration, transfer of an alkyl group from one molecule to another. Alkyl groups example ethyl, methyl, propyl, etc. Alkylating agents are monofunctional agents such as temozolomide, then bifunctional agents such as chlorambucil and cyclophosphamide. Most vulnerable site of alkylation is oxygen of 6-carbon atom of guanine. Product of methylation of guanine is oxys-methylguanine. O6-methylguanine mispairs with thymine instead of cytosine. Base analog incorporation. Base analogs are molecules that can substitute for normal bases in nucleic acid. Substitution of a base analog will result in altered base pairings and structural changes that affect DNA replication and transcription of genes. Example of base analogs are 5-bromouracil, 2-aminopurine, 6-mercaptopurine, acycloguanosine. The structure of normal base and base analog and the base pairing, normal base pairing and mispairing is shown here. The mechanism of base analog incorporation. Mismatched bases. Mismatched bases are incorporated during DNA replication. Many such removed by proofreading mechanism. One that missed by it cause change in bases. Repaired by mismatch repair mechanism. Double base alterations. First one is pyrimidine dimer. It is a double base alteration. These are molecular lesions formed from thymine or cytosine bases in DNA via photochemical reactions. Cause effects of UV radiation. UV induces the formation of covalent linkage between consecutive bases along the nucleotide chain. Formation of thymine dimer lesion in DNA. The photon causes two consecutive bases on one strand to bind together, destroying the normal base pairing double strand structure in that area. UV exposure and formation of thymine dimer. The second one is purine dimer. It is also a double base alteration. Purine dimers are formed by oxidation of DNA. Causes effects of UV radiation. These comprise the photoproducts that involve at least one adenine residue that undergoes photocycloaddition reaction with contiguous adenine or thymine. Structure of purinic photoproduct, that is adenine dimer, post-K photoproduct and thymine adenine photoadduct. Chain breaks. Single strand break. Discontinuities in one strand of the DNA double helix. Chemical structure is altered in one strand. Cause mainly by irradiation and free radicals. Repaired by homologous end joining. Double strand break. Chemical structure is altered in both strands. Cause mainly by irradiation and free radical injury. Ionizing radiation induced damage. Ionizing radiation may damage the DNA directly or indirectly. Directly, it causes double strand or single strand break and it may lead to cell death. Indirectly, the free radicals and oxygen reactive oxygen species may cause double strand break and single strand break and it further lead to cell death. Difference between single strand break and double strand break. Single strand break in DNA damage that occurs in one strand of the DNA double helix. In case of double strand break, DNA damage that occurs in both strands of the DNA double helix. Single strand break are very common, but double strand breaks are comparatively rare. Single strand break can be easily repaired by the cellular repair mechanism, but double strand break cannot be easily repaired by the cellular repair mechanisms. Single strand breaks are not lethal, but double strand breaks are lethal since they cause different diseases. Cross-linking between DNA and DNA. Cross-linking of DNA occurs when various exogenous or endogenous agents react with two nucleotides of DNA, forming a covalent linkage between them. This cross-linking can occur within the same strand, intrastrand, or between opposite strands of double-stranded DNA, interstrand. These adducts interfere with cellular metabolism such as DNA replication and transcription, triggering cell death. Interstrand and intrastrand cross-linking is shown here. Cross-linking between DNA and protein. DNA protein cross-links are bulky DNA lesions formed when proteins become covalently trapped on chromosomal DNA. DNA protein cross-linkings can be induced by exposure to various physical and chemical agents including ionizing radiation, UV light, transition metal ions of nickel and chromium, environmental contaminants, and common anti-cancer drugs such as nitrogen mustards, mitomycin C, and platinum compounds. Chemical structure of DNA protein cross-links induced by reactive oxygen species. The structural representation is shown here. That's it. Thank you.